Okay, everybody should be gathering from the old YouTube link to the new one that I just put in the chat. So it'll be just a second, Anna, while people make
Okay,
the
so you
migration.
posted that on YouTube?
Yeah. And it looks like people are filtering over. And okay. All right, why don't we get started? It looks like just about everybody who was at the old link has made the transition over to the new one. Craig, thanks very much for your help. Good morning, good afternoon, everyone. Thank you for joining us live from the seafloor in the Gulf of California. My name is Ken Costell. I'm the Director of Research Communications at the Woods Hole Oceanographic Institution. And I'm not on the seafloor or in the Gulf of California, as you may be able to tell. I'm in sunny but cold Cape Cod. But thanks to the broadband satellite connection, we're going to talk to a group of folks on the research vessel Roger Revelle, which is operated by our friends and colleagues at the Scripps Institution of Oceanography. They're in the Gulf of California on a 3-week research expedition to study hydrothermal vents on the seafloor in a region known as the Guaymas Basin. And they're using the remotely operated vehicle Jason and the autonomous underwater vehicle Sentry to do that. We'll hear about what they're doing on the ship and what they're learning with the vehicles in a moment. First, I just want to introduce to you the chief scientist of this National Science Foundation funded expedition. Dr. Anna Michelle is an engineer and associate scientist at WHOI in the Department of Applied Ocean Physics and Engineering. She specializes in the development of what are known as in situ sensors, optical sensors. Those are instruments designed to withstand the conditions of a particular location or environment to measure things like methane in this case, or microplastics. And in this case, it would also mean being able to withstand the crushing pressure, near freezing temperature, and complex chemistry of the seawater around a hydrothermal vent. Dr. Michelle, thanks for being here. I know you want to turn things over to the scientists or to the students that you have on board the ship, but I was wondering first if you just kick things off by telling us a little bit more about where you are, what you're doing.
Great, thank you, Ken, and thank you everybody for joining us today. So what you're seeing is a live exploration at Guaymas Basin. So we are at a site that we've named Chimney 1. You can see the black smoker and you can see a lot of tube worms around it. So we are live from the Jason van, and we have engineers here from the National Deep Submergence Facility who are operating the ROV today. So they'll be moving the cameras around, moving the vehicle around during today's show. We're also joined here in the van by a group of students, engineers, faculty who are here to study this environment. We brought out an interdisciplinary team, so we've got biologists, we've got geologists, we've got engineers, we've got chemists. I probably missed somebody's expertise, but we've pretty much got somebody who can answer any questions today you might have on what you're seeing. We're going to have today's live stream hosted by 3 different graduate students. So we've got Karina, who will tell us a little bit about herself in a minute, who is joining us from Mexico. She's from CICESE. We've got Yuna, who's a graduate student at Harvard University, who can tell us about the biology she's studying. And we've got Brooke, who's also from Harvard, who's going to tell us about the tube worms today. So I'm going to turn it over to them. What you'll see today is you'll see us exploring these chimneys. We'll hopefully see some tube worms. At the same time, we have a suite of instruments that we're using to measure the chemistry of these environments. Some of these instruments have never been used before on the seafloor, so we're using these dives to really test them and see how we can develop them into even better instruments. So I'm going to turn it over to the graduate students who are here, and maybe they can introduce themselves. So you want to go ahead, Karina? And Karina will also be translating some of it into Spanish today, so hopefully our audience from Mexico who's joining us will also get to learn a lot from Karina.
Hi everyone, I'm Karina Fuentes. I'm from CICESE in Ensenada, California, Mexico. I'm a PhD student and I'm studying Earth Sciences. Hola a todos, mi nombre es Karina Fuentes y soy de Ensenada, Baja California, México. Estudio en CICESE y estudio Ciencias de la Tierra. Y bueno, aquí estamos estudiando más que nada, mi parte es la parte geológica y la alteración que se produce en este lugar, sobre todo en las rocas y en el suelo. A toda la audiencia hispanohablante que quiera hacer preguntas en español, siéntanse libres de hacerlo, que con gusto vamos a estar respondiendo. Muchas gracias.
Hi everyone, my name is Yuna and I study microbial evolution. And on this cruise, I'm going to be studying, it's collecting samples from waters, plume waters from these underwater volcanoes and microbial mats. Hopefully we'll get to see some. And yeah, I'm part of the Gurgis lab at Harvard University.
Thanks, Yuna. And I'm Brooke Travis. I am a first-year graduate student in the Gurgis lab at Harvard University, where I study deep sea biology with a focus on symbiotic relationships. On this cruise, I'm really interested in Riftia, as you see right in front of you, those little tube worms that Corey is zooming in on. And I'm interested in using the lens of symbiosis to answer questions about their metabolic characteristics. So all three of us are here and happy to answer your questions, and look forward to engaging with you guys.
Well, why don't we go around the horn and to all three of you and tell us a little bit more about what each of you is studying on this particular expedition and how it meshes with what you're working on more extensively back on shore.
Bueno, voy a responder primeramente en español. Como estudiante de Ciencias de la Tierra, lo que más nos interesa son los procesos y las alteraciones geológicas que ocurren en este tipo de ambientes, sobre todo los parámetros físicos que afectan la roca, el suelo, y con eso podemos generar modelos que nos ayuden a explicar todos estos alteraciones que están ocurriendo, que a veces no podemos realmente ver más que estas estructuras por encima del suelo marino, pero por debajo están ocurriendo un montón de cosas que gracias a las mediciones físicas que podemos hacer del suelo podemos generar modelos y podemos entender todos estos procesos que ocurren debajo del suelo marino para que podamos ver esto por encima del suelo marino.
So I study microbial genomes. So what I would do with these samples would be I collect samples from these environments and I extract DNA, and then I sequence the DNA. And then I analyze the microbial genomes. So what I'm interested in understanding is how microbes adapt across very diverse environments. So these hydrothermal environments are very cool because we have these very drastic gradients of geochemistry, and I'm trying to understand how microbial populations adapt to different gradients across these extreme environments.
Thanks, Yuna. And this is Brooke talking, and I am once again interested in the Riftia tube worms that you see present in front of you. And like you and me, they can also breathe and create energy. In humans, we use oxygen to do that. And by creating energy, it's through a three-step process. One of those processes is actually something called the Calvin-Benson cycle. And that is something we'd like to investigate in Riftia because they can also do that with the use of their endosymbionts, which means symbionts that are located within them. These symbionts can also do something really cool that we cannot, called the reverse citric acid cycle. So our questions on this cruise are, how are they doing this? What are the rates of that the symbionts are using RTCA, reverse citric acid cycle, and the Calvin-Benson cycle. So hopefully we'll be able to answer some of those questions and learn some new things.
Yuna, you mentioned that you're looking at what occurs across the gradients that are present in this near hydrothermal vents. How fast or how drastically can different conditions change, like temperature or chemical composition around hydrothermal vents? What kind of space are you talking about?
Yeah, so when we take temperature from the orifice of the vent, that can reach up to 350 degrees Celsius. But then right outside of it, about a meter away, would be 2 degrees. So that's the sort of gradient that we're talking about here. But also I'm looking at temporal changes. So some of these microbial mats appear, and then when we go back to it 2 years later or a year later, they're gone. So we can sort of understand how these features are not stationary. It's very dynamic. And we find new chimneys, old chimneys, dead chimneys, and so on. So these environments or these habitats are not, these habitats are very dynamic environments.
So not only over long periods of time can chimneys appear and disappear, but even over the period of minute to minute, things can change very drastically for some of the organisms that are living there.
Yes. So that's what one of the things that we're trying to capture here with the sensors. With the in situ sensors, we are able to really grasp what kind of minerals, what kind of gases are coming out of these vents using tools such as mass spec, we call them. So we can analyze the different kind of gases that come out as it happens.
And Karina, I don't know if you see in the chat, there was a question that I pasted from YouTube in Spanish, if you want to field that one.
Yeah, hi, I see it. Ok, para responder la pregunta, bueno, estamos también aquí en el Golfo de California como en la expedición anterior. Entonces estamos hablando de ambientes similares en el que magma asciende cerca de la superficie del suelo marino, debido a los procesos de la apertura del Golfo de California. Sin embargo, lo que se diferencia este sitio, que es la cuenca Guaymas, con el sitio anterior, que era la cuenca Pescador, es las distintas etapas de maduración, por así decirlo, debido al esparcimiento del suelo marino. Lo que nos interesa aquí en Guaymas es precisamente que podemos estudiar básicamente, por decirlo de alguna forma, en tiempo real, todos los procesos principalmente petrofísicos que están ocurriendo gracias a las intrusiones de esta ascendencia del magma debido, como menciono de nuevo, a la apertura del Golfo de California.
And could I ask you to translate the question into English and give us an English explanation of what you just said? It doesn't have to be as in-depth if you don't want.
Yeah, of course, I try to do my best. Okay, the question was how this expedition can relate to the past expedition that were in Pescadero Basin. So I was talking about both expedition were here in the Gulf of California, but the difference between the expeditions is the study of the evolution of the process that are related to the seafloor spreading. Here in Cuenca Guaymas, Guaymas Basin, the evolution, it can be more like the maturity of this basin is like we are studying.
Thank you for that. I think Brooke, there's another. Oh wait, before we go to you, Brooke. Well, this might be for you as well. We had a couple of questions asking how deep is Jason operating right now?
Yeah, so right now the depth currently is 1,808 meters, 1,800 meters basically, below sea level.
So Brooke, you mentioned that the tube worms have endosymbionts, these symbiotic bacteria inside them that help support their life cycle. And I was wondering if you could explain that a little bit more and tell us what is the citric, you were talking about the reverse citric acid cycle. what is it that exactly that the bacteria do to help keep tube worms alive?
Yeah, that's a great question. So let me back up and just explain what a symbiotic relationship is. A symbiotic relationship is defined as two organisms living closely together where they each gain something. So in this case, these tube worms here are these red kind of worm structures that you see in front of you. So that's one end of the symbiotic relationship. And the other end are these gamma proteobacteria that actually live inside them. So that relationship is what I'm interested in studying. And one example of how they help each other would be Riftia kind of gains the nutrients from the surrounding water. You can see right here, the red kind of fluffy structure is actually the gills of the tube worms. So that's where they're breathing and gaining nutrients from the outside environment. Some of that nutrition is given to their bacteria that live inside them. In exchange, the bacteria can actually utilize some nutrients that most animals can't in the surrounding water, and in turn help the Riftia grow and thrive that way. So as you can see, it's a very intricate and interesting relationship.
I don't know which of you might be able to answer this, but now these hydrothermal vents are somewhat special in a way that they're not all, well, the fluid is not entirely passing through rock. They're also passing through a lot of sediment, correct?
Yeah, they would be called seeps as well. So we can see like little bubbles that come out of the sediments. That would be one of the features.
So what does that do to the chemistry of this fluid that's coming up?
So there will be dissolved gases in these fluids, and then they might manifest as they come up as bubbles. So we would often see microbial mat features in these seeps because there are all these chemicals that microbes can use to gain energy. So these would be hotspots for biodiversity and microbial activity.
Yeah, in addition, let me add one thing. The tube worms that you see in front of you can actually change the pattern of the flow of the gases and heat that are coming out of the chimneys. We call that diffuse flow. So essentially, because that they're physically present, the water kind of has to find interesting ways of seeping out of the rock and material. So in addition, like Una was saying, the Riftia can also change the chemistry of the chemicals that are coming out.
And are there relationships between the different organisms that you see on a vent?
Yeah, definitely. So here right in front of you, I can tell you the red kind of scaly worms that are crawling on the outside of the tubes. Those are called scale worms. And to be honest, we're not entirely sure how they're related to the tube worms in this environment. But a theory is they could possibly be a source of other bacteria and symbionts or even parasites. So, but that is yet to be explored. So a lot of questions we're asking today.
There's always a lot of questions. I do want to turn to a little bit of the human side of exploration. We have a lot of people who are obviously friends and family of your all's checking in on YouTube and saying hello. And I know that research happens around the clock and throughout the calendar. So all of you who celebrate Thanksgiving are going to be missing it. How do you plan to connect with family, or how are you going to be celebrating the holiday on the ship?
Yeah, so we're gonna be having a big meal tomorrow. So
Having been on research ships before, I know that's not.
Meals are great
special.
usually.
Yeah.
Yeah.
Well,
Tomorrow it's gonna be extra special. We might have bad weather though, so that might be exciting. Just to switch until now, we've been having amazing weather for research. Everything, the seas are calm. And as scientists who work in the lab, it's really lucky to have calm water so that things don't fly everywhere and we can do everything with calmness without strapping things down. So tomorrow, yeah, we'll have a big meal, and yeah, we'll continue working because we have operations running almost all the time. Yeah.
Somebody asked big meal of worms. No, you do not want to eat one of these things. You know, you mentioned that that there's, you know, a lot of work going on in the lab. So that reminds me that, you know, what we're seeing on the screen of all of the work that's going on the seafloor is only part of what's happening on the research expedition. Tell us a little bit about what kinds of samples are being brought to the surface and what you're able to do with them in the various lab spaces on the ship.
Yeah, so this is Brooke. I'll just say a little bit about the biology samples. So what we do to grab these tube worms in front of you is Jason has some arms, actually, like robotic arms that the pilots will use to manipulate and grab some of these worms off the rock. And on the ROV, we actually have a box that we just put them in until Jason returns to the surface. So I'm collecting a lot of tube worms, and we get a few other little critters in there as well, like some other scale worms and other small worms here and there. But in addition, we've been collecting numerous other things, including actual samples of the chimneys that you see in front of you. So the rocks and things like that for our colleagues here on the ship. In addition, you can talk a little bit about the samples, and then we'll pass it off to Karina.
Yeah, sure. So Jason does a lot of things. So I don't know if we can see the push cores. Is it possible to show push cores at all?
Here, we'll look at that.
Yeah. So push cores are basically one of these cylinders that we can press into the sediment and then bring up the whole core of a sediment. So I'll be collecting these in microbial mats and then processing them in the lab. So what I would do is collect relevant sections of the core and then flash freeze them in liquid nitrogen so that we can preserve the nucleic acid or DNA material, so for me to process back at home. And another big sort of operation that we do here is Ctd Casts. So if we basically bring down or send down cylinders and that to collect water at different depths, and we've been using that to really collect plume water. Plume is this volcanic or underwater volcanic infused volcano material infused water that's been that we can find in the water column. And I've been filtering this water to collect the microbial fraction, and I'll be sequencing the DNA from the filters as well. Is there anything else?
The major.
So this is what we see on the screen here is called major sampler. What's cool about this particular sampling is we can collect really hot fluids without really melting. This is made out of metal that can collect very hot fluids. So this is used to really characterize the plume water or volcanic water that comes right out of the orifice of the chimneys in order to characterize the source fluid.
With the major samplers, do those samples come up at pressure?
No.
Okay.
No. As the Jason ROV ascends to the surface, everything is pretty much under the atmospheric pressure that we are living under, which is 1 atmosphere.
Okay. Sorry, I think I might have cut somebody off at the very end there.
Yeah, I'll let Karina speak here a little bit about her sampling.
Okay.
Hi. We also are acquiring heat flow measurements. That is a physical parameter that can help us to understand the plumbing system of heat flow and hydrothermal flow that occurs below the sediments. So, en español también estamos utilizando datos físicos como la medición de flujo de calor, que nos permite entender como la plomería que está ocurriendo debajo de los sedimentos, que se da por los flujos hidrotermales, que son estos flujos de agua caliente que van alterando los sedimentos. Thanks.
And I would like to add that we also have autonomous vehicle called the Sentry operating at the sometimes at the same time, at different times to Jason, and there's a whole team of engineers working on that in order to collect data autonomously with this amazing robot that I don't know if we can't show photos at this point, but yeah, it's an amazing tool to study underwater features. And on top of that, Jason has all these sensors that we're testing currently, including pH and temperature, and also DIC, dissolved inorganic carbon, and we can sniff methane. So these are very useful tools for us to identify and navigate the underwater environment.
I am trying to find a picture of Sentry that
Oh yeah.
I can share. Only the hosts.
This,
So,
what we're
yeah.
seeing here is a sampling wand that has temperature and does it have pH? Is that the pH? No. No. Okay. Yeah.
Yes, it is. Yes,
It is also pH, and also a pump that collects water. Yeah. Oh, that's the Sentry.
There you go. Our flying barb soap. No, I say that with all love. I have a soft spot for Sentry. Tell us what Sentry is doing. This is for those of you who are just tuning in. I should rewind a little bit. We're live from the Gulf of California on the research vessel Roger Revelle, with a team of scientists and students from Hui, from Harvard, Michigan. I think Michigan State is represented on there. Katrina, I can't remember where you hail from.
Karina's from Sesese.
Okay, thank you. And we're talking to them about the hydrothermal vents in the Guaymas Basin on the seafloor, 1,800 meters below the surface that they're exploring. I'm showing on the screen right now the autonomous underwater vehicle Sentry, a programmable robot that they put over the side to survey the seafloor. And I was wondering if you folks on the ship, if you could tell us a little bit more about what Sentry is doing on this mission.
Hi, Ken, I'll jump in here. So Sentry is actually out right now conducting a mission. And so what we have Sentry doing is we're really doing some chemical mapping of the area. So you can see, I think you can probably still see the live stream where you saw it before where the black smokers were coming up. So a lot of chemical changes are happening and that plume is rising up. And what we're doing is having Sentry go out and see if it can find that plume, but from a higher altitude in the sea, in the water. And so we actually are testing some new instruments on a Sentry. So we're having Sentry go out with these new instruments to see if we could actually look and find these chemical signatures. This is a great place to be doing this because we have this constant plumes that are coming up and that we can measure. But we're doing this as part of a longer scale idea that we want to be able to use Sentry and these new instruments to be actually able to go out and do underwater exploration and find things by sending out autonomous vehicles. So Sentry went down this morning about 6:30 this morning and is out for about a 24-hour mission. So Sentry will be back on deck tomorrow. And what's really cool is we can actually see the data. The Sentry sends us back little messages and we can actually see what Sentry is doing and what our instruments are doing while Sentry is diving. So we actually have 2 vehicles in the water right now. We have Sentry and Jason in the water. They're working in different locations. We keep a constant eye on making sure that the vehicles are nowhere near each other. But it's been working great to have both vehicles in the water.
Now, so if you're hunting, you're mapping the plume structure in the water, are you looking for new places of hydrothermal activity, or are you looking at existing locations?
So we know where we are here, but the idea is long term we would send it out and look for new areas. Here we're using it as sort of a test case, using our instruments to see what we can actually detect and how well we can detect them. Okay, so that gets me to a question from YouTube from Allison saying,
She's asking, have you seen any interesting or surprising organisms on the seafloor? And I was wondering if we could expand that just in general to the expedition. Have you learned anything new about this location or about these types of environments?
I'll let the biologists talk about what they've seen, but I think in terms of the mapping work we've done, we're actually pretty impressed on every dive. How we can see the plume layer, we can see the smoke layers we come down through the water column over a very large area. So that's been very interesting for us to see. We've also been very pleasantly surprised at how well our systems are working on Sentry and how we're able to do this chemical mapping. It's actually working extremely well. So we've all been very happy about that. I'm going to let the biologists answer a little bit about what they are seeing. Our biologists, several of them, this is their first cruise as graduate students, so I think they've all been amazed at what they've been seeing. So maybe you guys can tell them a little bit about what you've been amazed at.
Yeah, I guess I'll go first here. So of course, it's always very exciting to see, stingrays or, larger fish on the cams here. But a lot of the times we're seeing, the deep sea floor where you really have to have a close eye to see the biology and larger animals that are hanging around. We have seen a lot of octopuses, so that has been very exciting for the crew here. But in terms of what we're learning about new species on the deep sea floor, a lot of that we can't truly say for certain that this is a new species or not without doing further analysis like DNA sampling and et cetera, et cetera. Mm-hmm.
Brooke, before you hand it off to the other two, we do have a question here from Stacy specifically about tube worms. She was asking, can they move or are they just moving with the flow of the water?
Yeah, hi Stacy.
The general move, the slow movement. I know they're sessile organisms.
Yeah, great question, Stacy. So tube worms, the actual length of a tube worm is about 1 meter long, which is about 3 feet. Although here you can see longer ones that seem to be about 2 meters or 6 feet in length. So the question is, how are they at the top of their tube if they're truly not that long? So these tube worms do something really interesting, where as they grow and develop, they set up these false bottoms within the white tubes that allow them to kind of sit up, almost like you're sitting up higher on a booster chair, so that they can reach the fluid flow as they grow. That's pretty much the only movement that they'll do throughout their whole lives, except for when they're larvae and trying to find a new place to live. Mm-hmm.
Okay, so now the question over to Katrina and Una about anything new or surprising you're learning about this site.
Yeah, so we've been seeing a lot of macros or animals at the bottom of the ocean. That's been very exciting to see as we do watch duties. I've been really amazed at how smelly the water can be. So we bring up and also rocks. We bring up all these organisms and rocks and sulfide rocks as well as plume water, and they smell really bad, like rotten eggs, because it's so full of sulfides. And also we've been measuring really high methane concentrations. So that's been really interesting and exciting, because that also means there might be a lot of cool biology happening and cool microbiology happening. Yeah.
Well, on the heat flow measurements that we are measuring, we kind of see that there are like clams fields that seems to be associated with high heat flow data. That's new for us, that we don't expect that, but it seems that there's like some time of correlation with these clam fields and high flow measurements. En español, estábamos comentando que ha habido específicamente en las mediciones de flujo de calor, parece que hay una relación entre ciertas parches de entre ciertas partes de almejas, parches de almejas y mediciones altas de flujo de calor.
Someone was asking, what's the chemical composition of the black smoke or fluid that we're seeing coming out of the chimneys there? What? Yeah, just that. What is that?
So there's a lot of different chemicals that are in those fluids, and you see it's forming those particulate matter. So you see that it's precipitating out. So we have a couple instruments. So I'm just going to talk about what we're doing. So we have an instrument called a mass spectrometer that the Gerges lab is running, and they are looking at lots of different chemicals. Hey, Pete, can you tell us what you see currently?
Yeah, hi folks. A mass spectrometer is a sensor that can detect gases of different kinds that are dissolved in seawater. It's really useful down here because it helps us understand how much sulfide and methane and hydrogen there is in the vent water. And those are the gases that the microbes use. The mass spec right now is sniffing on the waters around the tube worms, and we're seeing lots of sulfide and lots of methane, lots of carbon dioxide, which is higher around these vents than it is in the ocean. So it's been working like a champ, and we're going to combine that with the data that Dr. Michelle's lab is collecting to understand the chemistry. So Anna, I'll turn it back to you to talk about your sensors.
Yeah, so we've got 3 different instruments that we're running. 2 of them are measuring methane, and so we're seeing very, very high levels of methane compared to the rest of the ocean, and also actually what we have in our atmosphere. We also have a new instrument for carbon dioxide that's measuring something called dissolved inorganic carbon. All of these instruments are measuring very, very high levels of it at this location. So we'll be combining all of this data later to look at what actually is coming out at this environment. We also run these instruments as we move around these areas to kind of see how these chemicals change both at the sites and just away from them.
Okay, I just want to do a quick rundown of some of the shout outs that we're getting from the comments. The Travis family is tuning in, the Pardis family. Una Brian says hello. Let's see.
Hi, Brian.
Katrina, Cecilia says your family is very proud of you.
Oh.
Let's see. Brooke, very proud of you, says Grammy.
Thank you, Grammy.
It's always nice to get words from family when you guys are out at sea.
Yeah,
So the 3 of you, Katrina, Una, and Brooke, you're all first time. This is your first expedition?
For me, it's like kind of the first. My first one was in Pescadero Basin about a month ago, so it was very near from here. But yeah, it has been pretty amazing to be in this research vessel. It has been very exciting for me.
I was wondering, I know we're going to be talking about sort of life on the research vessel on Monday, but I was wondering if you could just give me, what you Katrina, you already did, but if Una and Brooke, if you could also just give folks your impressions of life and work at sea.
Sure, yeah, I can go. Okay, so the schedule is kind of very strange because we're operating according to the schedule of Jason mostly and the Ctd casting. So I have 4 a.m. watches, so 4 to 8 a.m. I sit in the Jason van with the pilots and log data. But also when the samples come up, there's a whole chunk of work that we do in the lab. So we have to nap whenever we can. So the sleep schedule is strange, but we have strict meal times that we stick to. So we nap between work and meals. And also interesting is we have a gym here. So that's really nice to have. So we get to work out when we have any downtime. And And we have ping pongs and board games and movie nights. So there is, space and opportunities to relax and just socialize as well.
Before you hand it over to Brooke, you mentioned that you sit a watch from 4 to 8 in the control van. Could you tell folks what it's like in the Jason van?
Yeah, so we have a lot of screens, about like 32 screens apparently. And then we have 3 folks from the Jason team operating the operating Jason, and we sit on the second row, the watchers, and there's a data logger. So I log data, and then Sarah, who sits next to me, who does the watch with me, she logs the videos, and we see if there are any important measurements that are happening, or if we see anything interesting, then we have to log that data. And then we also have a third row of anyone who wants to watch, sit on the watch, and observe can also do that. But it's the van itself is basically 2 container shipping. We kind of made it so that it's a very dark space with a lot of screens, so we can watch what's going on. And it's very cold inside the van, even though we're in crystal waters. So at 4 a.m. I come in with a lot of jackets and blankets and coffee, and that's a very interesting experience. Yeah.
I just got off of Atlantis in Puerto Rico, and everybody else was wearing shorts and a t-shirt, and I was wearing long pants and a hoodie.
Yeah.
The whole time. For those of you watching on YouTube, I put up a shot of from inside the Jason van that was taken by somebody who I just noticed is tuning in. Ronnie Wims is a member of the Jason team, former bosun on Atlantis. He's watching from, I can't remember, South Carolina, North Carolina, one of the Carolinas. But this was taken of Corey Verheyen, who's currently piloting the Jason. He's down at the bottom of that photo. Sorry, Brooke, over to you about what you've been experiencing at sea.
Yeah, thanks for that. I will say the first few days I was a little seasick, so I was a little down for the count on that one. But once I started to feel better, I think the thing that surprised me the most was how much I'm able to learn from my amazing colleagues on ship. Because like Anna was saying previously, we have a lot of different specialties on board here. We have biologists, chemists, engineers, we have crew members, amazing cooks. So it's been great that every gathering I get to see them, I learn something new.
So we're going to be wrapping up here shortly, but I just wanted to let's go back to the live feed. We're playing it on the screen, of course, but I was wondering if you could tell us what specifically Jason is doing at this moment. What's this particular dive about?
Sure, I'll jump in there. So what we're doing right now is you can actually see the wand out, and it's actually sitting in a small little area where we're pumping fluids back to these instruments. So we have 4 different chemical sensors running. So we're measuring the methane, the carbon dioxide, and again, the mass spectrometer is measuring a variety of different gases. So we'll be doing that for a while. Then there'll be some tube worm collections later. We'll be doing some photo imagery of these sites. We're working right now at Chimney 1, which is kind of in the south part of this area. And later today, we'll be moving a bit north to check out some of the areas we haven't been looking at. So again, we'll be kind of looking at this from a interdisciplinary aspect. We'll be doing chemistry, the biology, the geology today.
And Anna, just before you hand it over, how hot are the fluids that we're seeing coming out of the vents?
So right now, the probe, which you can kind of see, is in a little orifice there, and the temperature we're reading is 7.8 degrees C.
And at the top end where it's really warm?
It's kind of a small area. I think maybe we can, Pete, could you zoom in on where we're measuring that temperature? These are not the hottest parts. If we stuck it into that black smoker over there, the other day we were reading, I think we were up to about 350. Unfortunately, when we stuck it in the other day, it stopped working. So I'm not 100% sure what temperature we got. It just kept going. But we've replaced it, and this one right here, now it's reading about 6 degrees C. But you can
So
see
it's
kind of its poke.
That's really sort of encapsulates that idea that Una was talking about these gradients. So we're going from 7 degrees,
And just
which is real warm. To
Yeah, and
really
if
hot.
we were to move this outside of this area, just into the regular seawater, it would be about 2 degrees C.
Okay.
You can actually see the fluid if you kind of look at that shimmery water, which hopefully you can see on your screen. I know it's a little bit hard to see, but you can kind of see it by the tube worm. That's the fluid coming out here that we're measuring.
And that's sort of like what when hydrothermal activity was first discovered, this is kind of what they saw. They just saw shimmering water. They didn't land on a black smoker. All right, well, we have hit the top of the hour, and I know you guys have more to do. Is there anything else you want to contribute before we sign off?
No, I think we'd like to let people know that we'll be live streaming again on Monday at the same time, and we'll be learning a little bit more about life at sea and what happens on the ship and who are all the people that make this expedition happen. So we hope people would join in again then. And we'd just like to thank everybody who watched today, whether or not you're friends or family or you're just interested. We're really, really excited that you joined us, and we hope you learned something today.
All right, thanks everyone. I'm Roger Revelle, and thank you to the audience who tuned in. Sorry about the technical difficulties to begin with, but as Dr. Michelle said, we'll be going live again at 1 o'clock Eastern on Monday, and we'll be learning more about what it's like to be at sea for 3 weeks on a research expedition. So whether you're an old hand or a brand new sailor or somebody who's never been to sea, you're sure to see things that either you recognize or will amaze you. Thanks again to the team on the research vessel Revelle. And I hope the weather isn't too bad for you tomorrow.
Hey, thanks, Ken, and happy
All right.
Thanksgiving to everybody.
And happy Thanksgiving to all of you.